Today we are going to talk about enzymes, which are proteins that act as biological catalysts. What does that mean? We're going to find out today. Before we get to enzymes, let's remember what a chemical reaction is. So remember, a chemical reaction is when reactants, that's what you start with, interact with each other to produce a new product. So that's what you end with. Sometimes you see those in chemical equations. And in a chemical equation, the reactants are always on the left side of the arrow, and the products are always on the right side of the arrow so that you know what you started with and what you end with. Another vocabulary word we're going to talk about here is catalyst. A catalyst is anything that lowers the activation energy that is needed to start a reaction. So to get a chemical reaction to start, you oftentimes need to have a little bit of energy input. It's like getting your car rolling from a stop. You give it a little bit of gas, and that's what starts the rolling. A catalyst lowers activation energy so that a reaction can get started. There's two major types of chemical reactions. There's endergonic reactions. They absorb free energy, and so when they occur, if you've ever touched one of those ice packs that gets cold when you break the insides open and it absorbs energy so it feels cool, that's an endergonic reaction. An exergonic reaction gives off energy, so it releases energy, and so that often is released as heat. And so in your notes, you're going to need to draw these graphs. The notes are a graph for an exergonic reaction and a graph for an endergonic reaction. The big things to notice are that in the exergonic reaction, it requires less energy to get it going, and the products are less energetic oftentimes than the reactants. And in an endergonic reaction, it takes more energy to get going, and the products are often more energetic than the reactants. An enzyme is an organic catalyst. So an enzyme is usually made of protein, and its job is to speed up chemical reactions that are happening inside of your body. Sometimes it's also referred to as an organic catalyst. Organic because it's made of the organic molecule, protein. Enzymes are also reusable, which means that once an enzyme has helped one chemical reaction happen, it can help another reaction happen if it's the same type of reaction. And enzymes act kind of like a lock and a key, and we'll talk about that in a minute. But their job is to lower activation energy needed to get a reaction started. Remember that activation energy is the energy needed to start a reaction. Like we talked about before, any energy that's put into the reaction, so that's this arrow here, that's the activation energy. Once you've activated the reaction, it takes less energy to keep it going. This graph shows us what a reaction looks like with an enzyme and without an enzyme. So with an enzyme, the energy required to get the reaction started is much higher than if you use an enzyme. That's why we say that enzymes lower activation energy. So they can get reactions started using less energy, which means that reactions can happen more quickly and more efficiently. When an enzyme is doing its job, it starts with a substrate. That's the name of the molecule. So in this image, you can see that the enzyme here is in gray. We're just going to label that E for enzyme. And the substrate is this molecule that's green. The substrate is going to bind to the enzyme, and the enzyme has an area called the active site. Here I'm highlighting it in red, and you'll notice the active site is the same shape as the substrate, so they can bind together. Then the product is what the enzyme does. So in this example, the enzyme is taking apart this molecule. So the product is these two molecules that are broken apart with the help of the enzyme. Again, a reactant is what you start with. So in this example, this is a chemical equation for making water. We start with our reactants, which are hydrogen molecules and oxygen molecules. When the reaction occurs, it gives us products. In this case, we get water molecules. Remember that the reactant is always on the right side of the arrow, and the product is always on the left side of the arrow. Enzymes act like a lock and a key. So the enzyme works in conjunction with the substrate to produce a product. So the enzyme is like the key, The substrate is like the lock, and the product is like the opening of a door once you've used the lock and the key together. Enzymes are also site-specific, meaning that there's only one enzyme per chemical reaction. So if you're always making water molecules, there's one kind of enzyme that can do that. There's another kind of enzyme that can break apart sugars. That enzyme couldn't make water molecules, but it could make many, many sugars. Enzymes can do two different things. They can break things down. That's called degradation. So you start with a molecule that's large, and you break it into two smaller pieces. Enzymes can also do something called synthesis, and that means they join two substrate molecules together. And so that means that you start with two different molecules with the substrate, and the product is one molecule. It's also important to know how enzymes are named. So enzymes end in the three letters A-S-E, so it's ACE is how you say that. And they're often named for the molecule that they act upon. So lactose, for example, is a sugar that is found in milk, and it is broken apart by an enzyme called lactase. Amylose is another sugar, like starch. It's broken apart by amylase. Lipids are fats, right? And they are broken apart by lipase. And hydrogen peroxide is broken apart by peroxidase. So that's how you name enzymes typically. Enzymes can stop functioning, and sometimes that's because the pH changes. So if an enzyme is used to functioning in a pH of 7, for example, and the pH changes to something that's lower than 7 or higher than 7, it changes the shape of the enzyme. That's called denaturing the enzyme. And that changes its shape so that it can no longer do its job. Temperature will also do that. So temperature also denatures enzymes. That's why, for example, when you cook a piece of chicken, it starts out pink and turns kind of white. Those proteins inside of the chicken are changing shape, and that includes the enzymes that are there. So here's a graph showing where enzymes are effective. So in a human body, our human body temperature is about 37 degrees Celsius, and you'll notice that about 37 degrees Celsius is where enzymes are most effective. If your body gets too cold or too hot, the enzymes stop functioning well. We also have enzymes in our bodies that are functional at different pHs. So for example, pepsin is an enzyme that's in your stomach, which is very acidic. So pepsin is most effective around a pH of 2 versus trypsin, which is found elsewhere in your body, and it is more effective at a pH of 7 or 8. So enzymes can be affected by both pH and temperature. When something denatures, the enzyme actually does change shape. So if the substrate normally looks like this, and the enzyme looks like this, at the right temperature, the two shapes fit together. The substrate doesn't change shape, but if the enzyme gets too hot and has been denatured, then the enzyme can change shape, and the active site, which is here, no longer can fit the substrate. And again, we see that with pH as well. If the enzyme active site changes shape, it's no longer complementary to the enzyme. Enzymes can also be slowed down if there is not enough substrate, right? So that means that there's not very many things for the enzyme to be active on, so the rate of reaction is not very fast. Another thing that can happen is that you have too little enzymes. You have lots and lots of substrate, but very little enzyme to go around, and that can slow down the rate of reaction as well.